Palmer thing is just like very upset that I'm not connected to it. Don't I have an intro slide on this? Do you guys have that?
No.
This is all I have?
Yeah.
Okay, we're off to a great start Monday morning. This thing, got to get rid of that Palmer thing. So where have we been? Gosh, it's like a virus or something, the Palmer Connect thing. Hide. My God. I refuse to connect to it. Well, I guess we're going to have to live with that thing. All right, so where have we been? So we completed fertilization. We created a zygote. That's kind of the definition. The beginning of life is after the zygote has been created. Then we split our one-celled zygote into a two-celled embryo. Remember, that's where the word embryo, there has to be two cells for embryo. And then about day 4, those daughter cells keep splitting and splitting. Remember, the daughter cells are called blastomeres in embryology. Once the cell becomes, or once the embryo becomes 16-cell stage, it's called morula. There was a little controversy on that, but we're going with 16. And then the morula segregated, still in about day 4, actually, where this occurred. And that's where some of the genes within the morula were turned on. Within the blastomeres were turned on. Nanog Wk3 woke up, and their gene products turned on Wnt, and the Wnt gene turned on nodal. We'll talk about this nodal gene over and over again. And with nodal on, and all the 4 of these on, the cells that have these 4 expressed, they clump into the center of our morula and form the inner cell mass, and we said that is us, the inner cell mass. Go away. All right. God, what in the... It's like... Does that happen to your computers too? All right, at least... see if this will... Maybe that will shut it up. All right, so we got the inner cell mass going on. The cells in the outer part of the pizza here, they express 2 different gene products. So 2 genes wake up, the CDX2 and the GATA3 gene woke up, and they inhibit the genes that would make Nanog and Wk3, so they keep them asleep. So without Nanog and Wk3 on, and as long as you're expressing CDX2 and GATA3, you're going to become this outer layer here. Better to call this the trophoblast layer, as opposed to the outer cell mass. Now that makes me, oh, there it is, trophoblast. I didn't fix that one. Some people call it the outer cell mass, but that's an AK for the trophoblast. So our little morula is kind of segregated. We're getting cells moving to different regions up there. All right, so now let's fall down to this rabbit hole. Just a little trip about the uterus, because we're going to talk about implantation. The uterus, this is an A to P view of it here. This is the vagina, which leads to this uterus. The uterus has 3 parts, which you'll actually see next quarter in Gross 2 anatomy. the fundus, the body, which is most of the uterus, is the body. The fundus is just the top of the uterus, and then the cervix right here. There's some fornixes, little kind of coves, that little outpouches that are here. This model didn't show, but you'll see that next quarter. The uterus opens, of course, to the vagina here, and then to the fallopian tubes. We've already kind of talked about the fallopian tube. The parts of the fallopian tube is the infundibulum is this area right at the end. The ampulla actually goes this entire length. This is all ampulla. And then the isthmus is this little piece right here. So that's the fallopian tube. Of course, that's hollow, we said. The uterus has some layers, which you'll get to, Dr. Doe will get to in histology. Has an outer layer called the perimetrium. Not very exciting. It has a muscular layer called the myometrium. What does myo mean? The word myo, muscle, like myocyte. Myo means muscle. And then it has an inner layer called the endometrium. Has two layers. I took all those slides out, too much histology stuff, but it's got two different layers. Stratum basale is the one we're really concerned with. That's where the implantation or the little embryo will get into that layer. But we'll just call it the endometrium for now. It is lined, it's important to know, it's lined with epithelial cells. Have you studied those yet? You just started. So here's another type of epithelial cell. Most of the cells are secretory, and they secrete a little juice. So the cavity of the uterus is kind of like, it has juice in it. And it's not empty. And then some of the endothelial cells actually have receptors on them, which could bind to a fertilized egg, and that's kind of an important clue. So that's where we're going. So about day 5, we've finally gotten out of day 4. By now we're about 36 cells, still called a morula. It actually drops into the uterus, kind of a martini glass if you're a martini drinker, kerplunk. And that's kind of a process, and I took most of those slides out as well, but there is a smooth muscle, kind of a sphincter around these fallopian tubes that it doesn't let things in and out real easily. But if toward the end of your period, the end of your cycle, you have your progesterone levels rise a little bit because of the corpus luteum secretes progesterone. And that makes these muscles kind of relax. So it allows our little morula to drop through here. Okay, so now we have a morula kind of floating around in the endometrial fluid. And interestingly enough, it's not, you would think it'd be watertight because of the zona pellucida, but it's actually getting pretty leaky. And here's in physiology, those of you who had physiology know about these sodium potassium ATPase pumps. Everybody heard of those before? Those are pretty common knowledge nowadays. Those genes are woken up by this point, and the blastamirs and even the zona pellucida are loaded with these things. And what they do is they start pulling sodium inside our little morula. And who follows sodium like a puppy dog? Water. Oh, not always. Physiology question. Can anybody give me an example where water and sodium move inside of a cell, not together? That's like a real easy physiology question. In the kidney region, it's got to do with antidiuretic hormone. Yeah, that causes the absorption of free water without sodium. So there are some exceptions to the rule, which you'll get to as you move through the program. But most of the time, if sodium goes inside the cell, water follows, and chloride often follows it as well. And so our morula is like a sinking ship. It starts to get loaded up with water. It doesn't sink, because it just floats around in there. But it fills with water so much that a cavity starts to form inside of our morula. And that, of course, has a name, and that's called the blastocyst cavity. Blastocyst cavity. That is such an important event that it actually changes the name from morula, now we go to a blastocyst. And that's going to be with us for quite a while. So we went from zygote to embryo to morula, and now we have a blastocyst. Those are like note card things. That's stuff you just need to know. Those are really easy type questions, high yield board stuff. All right, so the process of forming this blastocyst cavity, it's kind of easy to remember that. It's called cavitation. Cavitation is the process of forming a cavity. All right, and that blastocyst cavity is going to be really important because that's going to turn into the primary and then the secondary yolk sac as we move along. Here's a real human embryo, and you can see we talked about the inner cell mass already. The trophoblast is around the outside. You can see the zona pellucida still hanging out there. What happened to the corona radiata? That's gone. That thing just degenerated away. It's really not that important. But remember we said those cells are kind of loose anyway? They're not very strong. So those are gone, but the zona pellucida is still hanging in there. But yeah, so that we can see all the parts, and there's the blastocyst cavity. So if I put this on a test, a picture of this, without the names on it, of course, you should be able to tell me, is this a morula? Is this an embryo? Is this a fetus? Or is this a blastocyst? So the best word would be blastocyst because we can see the blastocyst cavity. All right, then there's the concept of these poles, the North Pole and the South Pole. Of course, it wouldn't be that easy. They give them different names, the embryonic pole and the abembryonic pole. The abembryonic pole is like the South Pole, the embryonic pole is like the North Pole. And so if we go back and look, we see that these cells have got stuck to one side of the trophoblast and not over here. And so for orientation purposes, this is called the embryonic pole right up here. And this is called the AB embryonic pole down here. Okay. Again, embryonic pole is up here. So the inner cell mass is stuck against the blank pole. That would be a question. Embryonic pole, abembryonic pole, the west pole, the east pole. I can put all sorts of crazy answers there, but it's the embryonic pole. And then the abembryonic pole is always 180 degrees away from the embryonic pole. All right, just another picture, kind of different look, but there's the inner cell mass, and that's you, right? This is going to be the placenta. That's you, is the first little human there. Nothing more to say about that. All right, the next big thing that happens, so we had cavitation, our little zygote formed a cavity in it. Now what needs to happen is we need to get away from that constricting, smothering zona pellucida. We cannot attach to the endometrial wall if that zona pellucida is like helicoptering around us. We need to break free. And so that's called hatching. So kind of toward the end of day 5, the hatching occurs. And the blastocyst literally hatches like an egg, like this. It literally hatches out of the zona pellucida. And because of the limitations on human embryology studies, we're not exactly sure how that happens. Let me go shut this door. Dr. Lou has that booming voice. So I bet you didn't know you hatched. We all hatched. All right, so we really don't know how this works. We know that the inner cell mass releases some enzymes, and it's so confusing. It's not in the embryology books. I'm not even going to go down that rabbit hole. But there's a whole bunch of enzymes that are released, and they literally bore a hole toward the North Pole, toward the embryonic pole is where hatching occurs. So that's something you should know. And it kind of rips up the zona pellucida to the point where this can squeeze out through all sorts of weird contractions, and we won't go down that rabbit hole, but it literally hatches out of the zona pellucida. And so now we have a naked blastocyst floating in the martini glass, and now it has the ability to bind to the endometrium. Okay? What happened to the corona radiata? We already said it degenerated away about day 2 while the embryo was still in the fallopian tube. That could be a little tougher question. Where does the corona radiata, where do we lose the corona radiata? Is it inside the uterine cavity? Is it inside the wall of the uterus? Or is it in the fallopian tubes? It's way back in the fallopian tubes. So we got rid of that one a long time ago. More segregation occurs. So now the inner cell mass itself starts to segregate into 2 parts. So we're going to get a layer that is more toward the embryonic pole. You can call it the outer layer if you want. And that's going to become something called the epiblast. And those are the most important cells of the human right here. These are super duper stem cells right here. They can become any type of tissue in the body. There's other super duper stem cells as well, which are created by these epiblast cells. These are for doing research and stuff. These are the ones that are most sought after. You can make a heart, you can make a pancreas, you can make an eyeball, you name it, you can make it out of these cells. Those are going to be located toward the embryonic pole. They're columnar in shape. I don't think I would ask that, but just to throw that out there. The layer that's underneath those, or more toward the south pole or abembryonic pole, that's going to become the hypoblast, which is made of hypoblast cells. So we've just created something called the bilaminar disc. So this is the bilaminar disc. So our inner cell mass is now segregated into a more dorsally located, or toward the embryonic pole, epiblast, group of epiblast cells, or a more ventrally located hypoblast. So double layered. And our blastocyst cavity, sometimes called the blastocele, has gotten much bigger. That ship is still leaking and taking on water. Okay, again, these epiblast cells and hypoblast cells are super, super powerful stem cells. That, of course, has a name. Those are called pluripotent. So they're pluripotent cells. And again, the epiblast cells are, there's not even a word for it, but they're like super pluripotent cells, the epiblast cells. They give rise to all the somatic cells in the body. Remember, we're made up of somatic cells and then some sex cells, the sperm and egg. They give rise to everything. And they'll eventually give rise to the 3 germ layers, which we'll get to when the time comes. So, oh no, there's the little nerdy guy. Here we go. Some more crazy stuff. How does this segregation occur? How do you become an epiblast cell? How do you become a hypoblast cell? Well, the new star of the show, and I don't think we know this one yet, there's another gene called the FGF4 gene, or the fibroblast growth factor gene. That gets turned on. They think it's nanog. Remember we talked about nanog and GATA4 are already on in some cells. I think it's that what induces it, but the key is FGF4 gene wakes up, runs the central dogma, and now you got an FGF protein floating around in your cell. And what does that do? That basically, without getting too deep in the weeds, that just causes the cells that have FGF4 turned on, it causes them to move up toward the embryonic pole. So those are going to become the epiblast cells. All right. All right, so what happens then? How come the other cells don't go up toward the embryonic pole? So FGF4 diffuses into the adjacent cells. So FGF4 gene is the first one to turn on. And so not only will it cause the cell that it's expressing to go up toward the top, become epiblast, it also soaks into the neighbor cells. And if the neighbor cells were a little slow and didn't turn on their FGF4 gene, the GATA6 will shut that gene off and block it so it won't turn on. And so if you can't turn on FGF4, you just get pushed to the bottom. So you'll become the hypoblast cell. So FGF4 is expressed in epiblast cells, and GATA6 is, well, I mean, that's kind of not the true, because no, that's true. GATA6 is expressed in hypoblast cells. We'll just keep it simple like that. Okay, but really, we'll just leave it like that. I'm not going to go. This is like crazy stuff. That's enough for you guys. All right, so here's a little cartoon of that. So the blue cells here, FGF4 woke up or turned on. We don't worry about what turned it on. And it causes them to migrate up here in clump. But they're also, the FGF4 soaks into these genes and turns on GATA6. And with GATA6 on, FGF4 can't wake up in these kind of yellow cells. So they get pushed down to the bottom, so we get this kind of look right here. Although, did I mess up the colors? Let's see. Yeah, I got them backwards. Yep, my amazing Photoshop skills failed us. But the FGF4 expressing cells are up here at the top. Hypoblast cells are on the bottom. GATA6 expressed here, FGF4 expressed here. Good? Mm-hmm. All right. And then that created, this is our bilaminar disc. And remember, we still, I keep showing these pictures. We have to remember this whole thing is a little ball of cells. I'm just showing you slices all the time, but this whole thing is a ball of cells. Right. All right, so bilaminar disc, we've just created a bilaminar disc. Sometimes it's called a bilaminar blastoderm, a bilateral disc with a C. Bilaminar embryonic disc, I think most people will just call it the bilaminar disc. But a bilaminar disc, you should know this a bit. Here's a softball, easy question. What is a bilaminar disc made of? It's made of epiblast and hypoblast. Seems simple right now, but epiblast cells are going to morph into ectoderm cells. Hypoblast cells are going to morph into endoderm cells, and we're going to form a mesoderm in the middle. Those are the three germ layers. So our bilaminar disc is going to turn into a trilaminar disc, and our trilaminar disc is not made of these hypoblast cells or epiblast cells. Those were the beginnings of that. So it seems easy here, but it's going to get a little more complex as we go. All right, we've also just established what's called the dorsal ventral axis of the embryo, because now we know which way is toward the back of the embryo and which way is toward the belly. The epiblast cells are on top, so we can always tell where the back of the little human would be. And the hypoblast cells are toward the bottom, so we now know up from down, or we know back from front in our little human. This would be the dorsal surface of the human right here, and this is going to be the ventral surface. Okay, that's pretty easy. All right, the AVE cells. Normally we take a break, but let's push a little further since we got such a late start. Another segregation story. These are important, these AVE cells. So this story occurs in the hypoblast cells only, specifically toward the cranial end of the hypoblast. And we don't even know which ways, we know the back and the bottom, but we don't know the cranial end from the caudal end yet. And this is going to show us which way the head will form, where these AVE cells. So in the hypoblast cells toward the cranial end, some genes wake up there And they're going to turn these cells into a very important cell type called the anterior visceral endoderm cells, or the AVE cells for short. So, oh no, here comes the little nerd again. So how do you make an AVE cell? What genes get turned on? It's the nodal gene must be turned off. And you would think, oh, that's really easy to do. That's not easy to do. That's a very dominant, powerful gene. And there's a bunch of genes that have to turn this thing off, which we'll look at here in a second. So to turn off the nodal gene, which is the first step of making AVE cells, you have to turn on a gene with a terrible name, Dickoff1, named after the science researcher who discovered. How'd you like that for a last name, Dickoff? You can think of all kinds of things to say about Dickoff if you're mad at him. But it's abbreviated often as DDK1 gene. So for the DDK1 gene, if that's turned on, you're probably going to be an AVE cell. All right, and so what does Dickoff1 do? It blocks the expression of Wnt gene. And we said last week that the Wnt gene actually turns on the nodal gene. It's really hard to turn off the nodal gene, so you have to go to the genes that control it. So you have to turn off Wnt. So DDK1 blocks Wnt. Without Wnt, the nodal will stop working. So this is such an important event to block that nodal gene that there's some helpers. In case maybe you have a mutation in the DDK1 gene, you have to turn it off there. So there's some backup genes that occur. There's like gangsters. I keep wanting to put a gangster. Lefty 1 and Cerebrus 1 sound like gangsters to me. But they're also turned on, and they also have the ability to turn off Wnt. So if there's a mutation in DDK1, it's okay because Lefty 1 or Cerebrus 1 could also turn off Wnt. So bottom line of this, DDK1, Lefty 1, and Cerebrus 1 or CERB1, they all have the ability to turn off nodal by turning off Wnt. So the board question could show, it could say it's going to turn off Wnt. Maybe it won't be so deep, and it'll just say it turns off nodal. So you should know both of those. Technically, it indirectly shuts off nodal, and it directly shuts off Wnt, but some authors don't go that deep, and I've seen board questions that don't go that deep. So, and by the way, these are hard board questions, right? These are for the top tier student. If you just want to get through this class, you can do so without knowing this stuff. But these are medium hard to hard type board questions. All right, so let's go back to our picture here. So we've already made the epiblast in blue, hypoblast in yellow here. Now these red cells right here, which used to be yellow, they started secreting DDK1, and that's going to turn into AVE cells. Once that happens, and scientists can tell this by studying what genes are turned on in these cells, now we know which way is going to be the head and which way is going to be the tail. So this is going to be the head. So this is your little brain and head right here, and this is the tail. So we haven't even implanted into mom yet, and we can already tell which way is the back, which way is the belly, which way is the head, and which way is the tail, and it hasn't even implanted yet. So molecular biologists have gotten pretty good at this stuff. And what is the job of the AVE cells? They are very, very important in making sure something called the primitive streak. Who's heard of that word, primitive streak? Nobody? Probably nobody back in the biology days of studying chickens or primitive streak is your spine and your spinal cord and your brain. It's going to be the start of that. So these AVE cells make sure that the primitive streak doesn't form over the top of them. It shuts off the cells that make the primitive streak above, and that'll make more sense as we study this further. If something's wrong with the AVE cells, plus they're for heart, the formation of the heart they're important with. If something goes wrong here, you're not going to make it. The fetus won't make it. Or if it does make it, there'll be horrible mutations and heart mutations, and it's really bad. So, How do the AVE cells inhibit the primitive streak? Pretty easy, actually. So remember the DDK1, Cerebrus 1, and Lefty 1, the nodal Turner-Offers? They secrete those three, and they float up and soak into the cells above them, and they turn off nodal. And so without nodal on, you can't form the primitive streak. It's really as simple as that. So no nodal, no primitive streak, and that forces the primitive streak to start down toward the tail, down caudally, where it's supposed to. So they inhibit nodal again. So kind of another nodal story. What else do these AVE cells do? This is an easy slide, a more typical board question slide. What do the AVE cells do besides inhibit the formation of the primitive streak? They're very important in forming the heart early on, the early heart structure, which we have to have. They're involved in that. They're also they're involved in something called the precordial plate formation. If you get more specific, the cranial AV cells form the heart, caudal AV cells form the precordial plate, and the precordial plate is part of the brain is formed from the precordial plate. So basically the heart and the brain come from these AVE cells. So they're really, really important cells. All right, so let's get this little floating thing attached to mom here. So the wall of the uterus, we said they're epithelial cells. It's really slippery. There's something that the epithelial cells secrete something called mucin 1, which that's what mucus is made of. Mucus is made of mucin plus water equals gross mucus. So you have basically mucus inside all the time, and the walls are very slippery. It would be hard to implant our little blastocyst if the walls are all slippery. You couldn't bind to it. So we need to shut this off, and that's what happens yet. So it down-regulates, and we won't get into all, we've done enough of those little nerdy guy stuff. We'll just keep it simple. The blastocyst secretes some stuff which turns off the slippery stuff, the mucin 1. And so basically the uterus starts to dry up, and now the binding sites are exposed. Okay, everything I said, I think. Did we go into the weeds? No, I did not go into the weeds. So with mucin 1 off, and now the epithelium kind of uncovered, not full of mucus, we can bind if we're lucky. So about day 7, this binding of our little embryo to the uterine wall occurs. And there's a couple receptors that you have to know for this. So on the blastocyst, remember that outer layer is called the trophoblast, we have something called L-selectins. So that's part of the binding, L-selectins. And then on the endometrial epithelial cells have receptors for the L-selectins called trophonins. There's a bunch of them. We'll just keep it easy. We'll just call them trophonins. There's like trophonin 6 and 7 and 8. And we'll just say trophonins. So L-selectins bind with trophonins. And here's exactly what happens. So here's our blastocyst. There's you, the little human. And here is a L-selectin on the trophoblast layer. And it finds the trophonin receptor for it on the endometrial cell. And the two bind and hook up. And that's a good thing. We're on our way to get into mom, so we can, because this is growing. We need blood, right? Right now, just the martini fluid. These things are drinking martini fluid and happy. But as this gets bigger, this thing is still growing and getting bigger. We have to get blood to this, or these cells are going to die. The only place blood is inside mom's endometrium. There's all sorts of blood vessels in there, and that's where we have to go to continue the growth of this thing. So if you have a mutation, here's like a good one of my question. A mutation in the blank receptor causes the blastocyst not to be able to bind to the endometrial wall. And so I could say a mutation in the L-selectin or gene, or a mutation in the trophonin gene. If you have a mutation in either one of those genes, you're not going to be able to get pregnant because you can't bind to mom's wall. So it could be the guy's fault too, may not be mom's fault. All right, ectopic pregnancy we should talk about. So that normally this binding occurs on the posterior wall of the body of the uterus and the uterine cavity. That's where it's supposed to occur. However, implantation can occur all over the place. Over 90% of the time, these ectopic pregnancies occur in the fallopian tubes. And that's not only will the baby not be able to make it there, it can kill mom because it can rupture the fallopian tube and you can bleed really, really bad from that. It's very, very painful. Mom will be into the hospital usually long before it ruptures. Maybe like if she's a drug addict on crack or something and she can't feel any pain, she could die from this thing rupturing. A normal person, the pain of the fallopian tube stretching is going to send mom to the hospital eventually. It's one of the causes of acute abdomen, like cholecystitis and appendicitis. Those will send you to the hospital because your belly is just killing you. We'll get into that more in 6th or 5th quarter. But yes, so let's look at this slide. Guaranteed question on this slide. So where does ectopic pregnancy occur? So 25% of the time in the isthmus of the fallopian tube. The most common place, so you should know this for sure. Don't memorize all this stuff, but know the most common place for it to occur is in the ampulla of the fallopian tube. There are receptors that can hook this thing up, and they're not the perfect receptors, but it can hook up here when it's not supposed to. 15% in the fimbriae, which is still kind of part of the fallopian tube. So really about 96% of the time, ectopic pregnancies are going to occur somewhere in the fallopian tube. Occasionally, once in a blue moon, remember we said sometimes the fimbriae will miss the egg and can't catch the egg, and it gets loose in mom's pelvic cavity, it can float around. So it can get stuck on the ovary, so a baby could grow, the sperm could come right out here and impregnate or fertilize this egg right on the ovary. It could get stuck anywhere in the pelvic cavity, in this rectouterine pouch or the pouch of Douglas, is about 1% occur here. And so it can occur in the pelvic cavity as well. It's rare. It can occur at the top of the fundus of the uterus about 2% of the time. Now these will probably be fine. There won't be much trouble here. Down in the cervix can be a little trouble sometimes. Baby can come out prematurely because it's going to be growing too close to mom's cervix. But it's pretty rare, 0.3% of the time. It's down here. So just know the big picture. Know that most of the time it occurs in the fallopian tube, and the most common spot is the ampulla, which is the main section of the fallopian tube. And it's possible for it to occur on the ovary. You don't have to memorize the number. It's possible for it to occur on the intestine within the pelvic cavity, anywhere in the pelvic cavity. It could be on the bladder. It could attach to that. It could come down over here and get stuck here. But those are really, really rare. So that's an ectopic pregnancy. I think you've probably heard that word before. Let's see. Should we take a break? Your brain's fried, or should we get through this and get out? Break? All right. No break? Break? All right. Let's take just a quick. Some people want a break, so let's do a little walk around break. All right, let's go. Let's get through this stuff.
That's me.
Another big word. How exciting. to start our break, end of break off. The syncytiotrophoblast. Syncytiotrophoblast. Sin City. Sin City. That's where DDK or Dick Off One lives. Dick Off One lives in syncytiotrophoblast or Sin City. All right, let's talk about some new cells. So we're implanted, we're bound to mom's outer endometrial lining now. That wakes up some of the trophoblast cells to start dividing by mitosis to make copies of themselves. And this is probably the strangest cell type you've ever met in your life. These new cells that are formed are called syncytiotrophoblast cells, and they are very, very important. They're very similar to cancer, strangely enough. So let's meet the syncytiotrophoblast cells. So very weird cells. Why are they weird? Because many of them completely lose their plasma membrane, and their cytoplasm morphs into the cytoplasm of their neighbor cell. They keep their nucleus, they keep their nuclear membrane, they keep their organelles, but they lose their plasma membrane, which is crazy. How can they survive? Don't know. They share mitochondria, they share Golgi tendon organs, but they form one big plasma membrane around all of them. So we have these big clusters of syncytiotrophoblast cells. So very, very strange. And they're very, very aggressive. They go through mitosis almost as fast as a cancer cell does. And somehow they know to go deep inside into the myometrium, into the wall of the uterus. They burrow. They're like burrowing cells. They burrow. We don't know how they know which way to go, but they do. And so so you get these big pack of these things, highly mitotic cells, very similar to invasive cancer. And the leading edge pushes them through the epithelial cells and deeper into the stroma of the endometrium. Let's look at a picture of the very first ones here. So here's the Martini glass out here, and here's the trophoblast layer, and some of these trophoblast cells have divided into these weird cells. Here's a cluster of 4 cells that have lost their cell membrane. They've kept their nuclei, and they kind of share a cell membrane. And these are dividing. The ones at the tip divide, and this gets bigger and bigger and bigger. As it gets bigger, and it doesn't happen over here on the trailing edge of the trophoblast, or our little creature. It only happens here where it contacted where it bound to the receptors on the endometrial cells. And so their job is to pull the embryo deeper and deeper into mom's wall. So how does it get through some of the cells? They release, and we'll just keep it really general. We'll get into the weeds in spinal anatomy on these MMPs. But we'll just call them matrix metalloproteases, which are very powerful enzymes which cut protein. So those leading cells also release these powerful proteolytic cutting enzymes. And they cut the mom's endothelial cells and fibroblasts. Any cells that they need to cut through, they can cut through these cells, and that's how they get in. So here's the cartoon. So they're releasing these MMP cells, and it cuts right through these simple columnar cells, and it starts to pull itself in. Okay, it also has some other chemicals that kill some cells, and I took most of those slides out. We don't need to go that far in the weeds, but very weird cell type. And it proliferates like a cancer. It gets bigger and bigger, and the bigger it gets, the easier it is to pull the little embryo deeper and deeper. They're also searching out mom's blood vessels, because mom has blood vessels in the uterus wall. And it's searching them out, and next week we'll look how they actually tap into mom's blood vessels and bring that blood into our growing embryo. So they're quite amazing cells. Now look at this one. So they've almost pulled the little embryo all the way inside mom's uterine wall. It's looking for these things. These are the blood vessels. So these will grow little points and jab into these. We'll see next week. But yeah, so it's pulled itself. It's gotten quite a bit bigger. It still hasn't started on the trailing edge yet. We don't know why that is. We don't know the mechanism of that. What else do these syncytiotrophoblasts cells do? They produce a very important pregnancy hormone called HCG, human chorionic gonadotropin. This is where HCG comes from. In fact, that's a fairly reliable way to see if you're pregnant or not. That's what these pregnancy tests use to see if the syncytiotrophoblasts are formed. Normally, you won't have syncytiotrophoblasts. They only form if implantation has occurred. So yeah. There's another job of HCG. I mean, that's not really a job to tell you if you're pregnant or not, but they extend the life of the corpus luteum. So after, we didn't, I cut that whole lecture out about oogenesis, but corpus luteum once contained the oocyte that hatched. So the egg that actually comes out of the ovary had a container, and it was called it was not the corpus luteum. Let me see, where's that picture of it? It's this, let's just go to this picture. So here's the little egg maturing, and there's hundreds of these things. It's a race to get to this stage, and only one will make it to this stage. This is called the Graafian follicle. The Graafian follicle will bind to the wall of the ovary and spit out the egg, or at this point it's a secondary oocyte. Remember, it's stuck in mitosis, or meiosis 2. But after it spits it out, it forms this donut structure, and that's called the corpus luteum, and that's secreting all sorts of hormones, but it dies real quickly. If HCG starts floating around in your bloodstream, it'll keep this thing alive for months, and that's good because this secretes progesterone and some estrogen, things we need to keep pregnancy going. So the HCG keeps the corpus luteum alive. It extends the life of the corpus luteum, and so it keeps so the corpus luteum secretes progesterone and estrogen. One of the trick questions is, do syncytiotrophoblasts cells secrete progesterone and estrogen? No, they don't. Maybe indirectly they do, because they keep the corpus luteum alive, which secretes the estrogen and progesterone. So watch out for that question. Okay, and then the placenta will actually eventually take over progesterone and estrogen secretion later on. All right, some name changes we have to do now. So we've been talking about that outer layer. It's been called the trophoblast. Once our blastocyst gets pulled inside the uterine wall, a name change occurs, and that's all it is. You don't call it the trophoblast anymore. Once it's inside the uterine wall, that outer layer of the embryo is called the cytotrophoblast. It's just an AKA. Cytotrophoblast is an AKA for trophoblast. Question?
Going back for a second, you said that like women, that's like a way to tell if they're pregnant or not with the secretion of the HCG.
Yes.
Do adult men have that naturally occurring at all?
No, unless they take steroids. Yep. Guys who take steroids can have trouble with that. Or actually, guys taking steroids sometimes have to get injections of HCG so they don't get breast formation. But normally you won't see that in guys. Good question. Okay, let's see. So, cytotrophoblast, and if you ever try to read this, nobody in the right mind is going to try to study embryology on their own. But if you ever wanted to and tried to read a book on it, the authors will drive you crazy because they keep going back and forth between cytotrophoblast and trophoblast. It took me forever to figure out. I had to go into the research and look at papers. What are these are AKAs for each other. All right. So now we call this cytotrophoblast, no more trophoblast. That's this whole layer here. And it's just a name change. Protection for mom. So what else does the blastocyst, or why doesn't the blastocyst get attacked by mom's immune system, right? because here's mom's blood supply. There's the microcapillaries. Mom has sensed that a foreign invader has entered her, right? Dad's genetic material is foreign to mom. Why doesn't this get attacked? And normally it would get attacked and destroyed. But the cytotrophoblast cells of the little embryo, they secrete a substance called EPF, early pregnancy factor. It's another thing that can be detected. Some pregnancy tests use this one. And we won't get into all the mechanisms, but it basically it forms a shield, kind of shield of this juice. EPF is kind of floating around here on the outside, and it kind of protects the embryo from attack from mom. It shuts off immunosurveillance. I won't get any deeper into that, but it protects from mom. We'll look at another mechanism next week as well. All right, another new structure has formed. About day 8, the amniotic cavity forms. So we'll take a look at that. By day 8, the syncytotrophoblast invasion has progressed quite a bit. And a new cavity forms between the cytotrophoblast and the epiblast cells. So let's take a look at that. So it's kind of toward the North Pole type thing. And which way is north? What pole is this right here? South Pole, or the embryonic pole. So now we can look inside. Here's the single layer of cytotrophoblast cells. And the author has actually they really shouldn't have done this. This remains a single layer. These should be all syncytotrophoblast cells. But between the cytotrophoblast cells and the inner cell mass, or the epiblast cells, we get a little cavity start to form here. And that's called the amniotic cavity. And that's going to produce amniotic fluid. You probably heard that word before. That's going to take over and eventually surround the entire little embryo. It's going to grow and grow and grow. But there's the first, about day 8 it first appears. Eventually we'll look like this. So here's the little bilaminar disc has turned into a kind of a human looking thing. But the amniotic cavity is huge, filled with amniotic fluid, which nourishes the baby and protects the baby, and it's very important stuff. Not studied, we don't know the story again because we're not allowed to study these things very much. And then it's got to have a layer around it, right? As it gets bigger, it's going to have its own layer. It's going to surround this. So let's look at that. That's called simply the amnion. The amnion walls off the amniotic cavity. So that's pretty easy. So these amnion cells, and in Europe they call them amnioblasts or amniogenic cells. We call them amnion cells, about the same thing. It is a very important layer. It's considered one of the extraembryonic membranes. I don't think that's terribly important though. Some authors, not for our boards, but they consider it extraembryonic mesoderm or ectoderm, which will make sense later on to you. But here's what happens. So these epiblast cells, and they're going to do this, and the hypoblast cells are going to do this. These ones on the ends go through mitosis and split. So if you split into 2 new cells, it creates a new cell here. So it's like that one didn't move, but it created a new cell here. And those are called amnion cells. So mitosis occurs again, and this one is pushed to here, and a new one forms here. They go through mitosis again, and these are all pushed forward. So pretty soon, this amniotic cavity is walled or lined by amnion cells, which secrete amniotic fluid, which is super important for human development. And yep, there's another picture of it. And enough, we have done it. Week 3 of embryology is over. Any questions? Online people? There's no online people. Well, they'll send me emails if they have questions. We're good? All right. Shutting down the shop.